So, continuing our discussion on organelle genome, we already in our previous interactions talked about mitochondrial genome, and now let us look at the other uh, genome, that is chloroplast genome. Now, both the genomes, as you saw, have similarities, and they have some subtle differences as well. Like mitochondrial DNA, chloroplast DNA is also double-stranded circular DNA, and it contains naked DNA, like mitochondrial DNA, in the sense that it is not associated with any protein like what you saw in nuclear DNA, which organizes itself in the form of chromatin. Now, that is this sharp difference in organelle DNA or the genome and the nuclear genome. So it's the naked DNA here. And as we discussed, the size of chloroplast DNA also varies from 85 kb to almost 2 kb, in different plant species, and it is larger than mitochondrial genome. The chloroplast genome is larger than the mitochondrial genome. And interestingly, each chloroplast has 10 to 60 copies of its genome. Like nuclear genome, you have single copy of all the genes, or the multiple copies are also there. But then in chloroplast, the same circular DNA is copied 10 to 60 times per chloroplast. And as you saw in mitochondrial genome also has multiple copies. So both the chloroplast genome and the mitochondrial genome exist in multiple copies, per organelle. Also, in chloroplast DNA, you have inverted repeats. The replication machinery is provided by nucleus in case of chloroplast. So whatever is required for DNA replication in chloroplast, that machinery is provided by the nucleus. And interestingly, as I mentioned, that genes of chloroplast, they have introns, and there are two categories of introns, one in the tRNA gene, which is in the anticodon loop, and the protein coding genes also have introns, like the nuclear gene have introns and exons. Similarly, chloroplast genes have introns and exons. And another sharp difference in mitochondrial DNA and the chloroplast DNA is that it is rich in GC contents. Okay, like for example, chloroplast DNA has 36% of GC content, nuclear DNA has 64, and mitochondrial DNA has 71% of GC content. So GC contents differ from DNA to DNA in the sense the organelle DNA, nuclear DNA, it has varied GC contents, and number of copies varies species to species. So these are some of the characteristics of chloroplast DNA. And the translation machinery of chloroplast is very much similar to the prokaryotes, and its genome contains genes responsible for rRNA, tRNA, some ribosomal proteins, RNA bundle subunits, translational factors, and the proteins involved in photosynthesis process. So it has a whole lot of genes, um, on on its genome. And like mitochondrial genome, chloroplast genome has also been sequenced, mapped, and annotated. This is the genomic map for rice or as a sativa. It's just to give you an idea that our knowledge today about the chloroplast genome has been up to date in terms of sequencing of this genome and mapping that genome, what all it contains. You know. Now, coming back to the ordinal inheritance. You know. So again, this aspect also has some history behind it, wherein as early as in the early part of last century, the first evidence of cytoplasmic inheritance was provided in terms of the variegated leaves, how that pattern is inherited from one generation to another, and around the mid part of the last century, the mitochondrial inheritance pattern was also reported for the cytoplasmic male sterility, which was observed in males. So these are some of the examples which have been later on studied, and we have a clear picture about how this inheritance takes place. Uh, so basically, ordinular inheritance, whether it is mitochondrial or chloroplast, it shows cytoplasmic inheritance, which deals with the traits which are coded by either chloroplast or mitochondrial genome. Okay, and these traits are then linked to their DNA. So that is the crux of the scenario. You know, so if we see the differences between the two inheritance patterns, the nuclear inheritance, which we now call Mendelian inheritance, and the ordinal inheritance, if we see. So Mendelian inheritance is governed by nuclear genes, whereas ordinal inheritance is governed by ordinal gene. And nuclear genes, they show distinct segregation pattern, whereas the ordinal genes, they do not exhibit segregation patterns the way shown by nuclear genes. Reciprocal differences are not observed in case of nuclear inheritance, whereas reciprocal differences are seen or observed in ordinal inheritance. I will elaborate this reciprocal differences in a while. Now, maternal effects are not seen in Mendelian inheritance, whereas it is seen clearly for ordinal inheritance. Genes can be easily mapped, and here mapping is difficult. And nuclear genes are associated with chromosome, whereas ordinal genes are associated with either chloroplast DNA or mitochondria. So these are some of the subtle differences in the inheritance shown by nuclear genes and the chloroplast genes and or the mitochondrial genes. Now, if you see this, how the ordinal inheritance uh, takes place. Now, if you see here, this is the reciprocal differences we were talking about, which is responsible for uniparental inheritance. So if you see here, this is the female gamete and this is the male gamete, and A, B, C, these are the nuclear genes, you know, and the male has the small a, small b, small c, and when these two cells fertilize, you know, the females has chloroplast and the mitochondria. Male also has chloroplast and the mitochondria. But when the two cells fuse, it is only the nuclear contents which go, not the cytoplasmic content. And you see the formation of the zygote, wherein you have the combination of a, small a, capital B, small b, capital C, small c, this. And the cytoplasmic contents are retained coming from female part. So it means whatever genes are coded by the organelles, they express themselves in this generation. Okay. Okay. So that is what happens. And if you change the partners, you know, take the male here and female here, then whatever is there in the organelles of this partner will come here, not not the other one. So here the inheritance or the cytoplasmic inheritance or organelle inheritance depend upon the female partner, not on the male partner. Now this is elaborated if you come here. I mean, or to conclude what I'm discussing here, the features of organelle inheritance, they lack segregation, okay, like nuclear genes show segregation, they do not show segregation. Irregular segregation in biparental inheritance, somatic segregation, association with organelle DNA, mutagenesis, yes, there are mutations, and lack of association with a parasite, symbiont, or virus. So, organelle DNA is free from these interactions from parasites or symbionts or viruses. They don't attack the organelle genome, whereas they affect the nuclear gene. Okay, so these are some of the key features of the organelle inheritance. Now, see the work done in 1908, which is historically first report of this kind. Now, if you look at this plant, which is known as 4 o'clock plant, you all must have seen this plant growing, and it is an ornamental plant grown at homes also with the name Mirabilis jalapa. Now, if you look at this leaf of this plant, these are three distinct types of leaves this plant has. Totally green leaves, or white leaves like this, and the variegate means green and white patches like this. Okay. So now this pattern of leaf was studied, you know, um, that what is responsible for this, whether it is nuclear coded phenomenon or something else is responsible. Okay, so what was seen here, so different kind of crosses were made. So if you take a plant with white leaves as female and green leaf as the male partner in the reproduction. So what you get here is all plants which has white leaves in the offspring. The green leaf doesn't come at all. If it was nuclear coded thing, then you should have seen 4:2 or 3:1 segregation, which is not happening. So you have white female plant and the green leaf male plant, cross it, and you see all offsprings have white leaves. When you reverse the parentage. You have female, which is has all green leaves, male, all white leaves. Cross them, and you see all offsprings have green leaves. Okay, so the trait of green leaf depend on the kind of female plant you are using here, whether it is white or green. So the leaf color depends on the on the female partner in the cross. You know. So similarly, if you come here, if you take variegated leaf plant as female and green as male and cross them, then what you see here is a combination of all three types of leaves. You have green leaf plants, white, and the variegated offsprings. So you have this kind of variation when you take this female, and similarly when you take female as green leaf partner and the variegated as male, in this recipe, in this cross, you get all green offsprings. Again, confirming that the it is the female partner which decides the leaf pattern of this plant, and that was the basis of the concept developed of cytoplasmic inheritance. It's not controlled by nuclear genes, because nuclear genes in these kind of crosses would have shown the normal Mendelian segregation pattern, but they are not showing. Here, the trait depends on the female phenotype, not the male phenotype. Now, how it happens? Let us look at this. So, for example, when you take a white female plant, so in their egg cells, the egg cell has these leucoplasts. Leucoplast means colorless plastids. So this is the thing. And when it is crossed with the male, even the green leaf male cell, you get this zygote where you have all the leukocytes, plastids, and you have all offspring plants have white leaves. When you take female which has green leaves, then you have these are the chloroplasts, and in the female cell you have the chloroplast, and when it fuses, forms a zygote, which is the transmission of these chloroplasts to the zygote, and all the offsprings have green leaves. Now, when you take the variegated leaf plant as the female partner, then you have the eggs which are of three types. You have eggs which have leucoplasts, and you have eggs which have only chloroplasts, and then you have the combination of leucoplast and chloroplast, both are present here. And when they have get fertilized with the male reproductive cell or the pollens, you have the zygote generated, three types of zygote. One is which has only leucoplast, only chloroplast, and both leucoplast and chloroplast. And when their offsprings, all will be white, all will be green, and then you will see this kind of variegated pattern. So, indicating very clearly that the variegation or pattern of leaf is guided by the cytoplasm or the presence of plastid, the type of plastid the female egg cell carries, decides whether the leaf is going to be white, green, or variegated. Now, this kind of study has been done in all sorts of plants, actually, after that. So, if we look at the mitochondrial inheritance, this is governed by genes located on mitochondrial genome. And one very good example, which has been studied and exploited for in plant breeding is the cytoplasmic male sterility. Cytoplasmic male sterility. It means the male sterility in plants is governed by cytoplasm, and that too by the mitochondrial genome, not by chloroplast genome. Okay, so now all these genes which are responsible for male sterility has been characterized, mapped, and the initial studies were done in sorghum and then in Neurospora fungus and yeast. And now we have examples from almost all economically important plants that this phenomenon is coded by mitochondrial genome. Okay, so to summarize as a whole, what we have done to look at the genome which is present in organelles like chloroplast and mitochondria, and what all they contain, and I have very specifically focused on the other traits than the functional genes which they contain, and they participate in the functions of respiration and photosynthesis. So I'm very specifically looking at the genome, the genes, and what particularly the genes which may be involved in trait modifications. Not the functional part of it. Okay? So as I mentioned, both the genomes have so many genes or many genes which help these organelles to perform their functions of respiration in mitochondria and photosynthesis. And so with this, I come to an end of this topic. And now we will look at a little bit in detail about the nuclear genome expression. That is how the nuclear genome expression is regulated. First, how it expresses itself and how it is regulated in a cell-specific manner or development stage-specific manner. Okay. So thank you for listening to the